Unbelievable. There's so much rubbish on the radio these days. Right, video. Hello, it's the sixth video in the evolutionary ecology series. Now, last time we learnt about some of the functional genes that have been identified and have been shown to be important in adaptive evolution. Now, all those traits that we looked at affected the morphology of the organism in some way. But we know that genes can influence a lot more things than morphology. They can influence behaviour as well. So that's what we're going to be looking at today. Now, a common idea surrounding the idea of adaptive behaviour is this, the extended phenotype, obviously a phrase popularised by Richard Dawkins. But what exactly does it mean? Well, it's actually quite simple, really. An extended phenotype is any functional entity constructed by an organism that isn't part of that organism. Okay, so things like birds' nests, the bowers that many fish produce to attract females, and also spiders' webs, where we've got a particularly good example. Okay, so Blackledge and Gillespie in 2004 was studying these orb-weaving spiders in Hawaii, particularly in the genus Tetragnatha. Okay, and in this genus there's a huge adaptive radiation on the islands of Hawaii. Now, one thing to take into account, on each of the Hawaiian islands, the conditions are relatively similar. Although many of the species of spider look quite similar, you can tell them apart more easily by looking at the webs they produce. So that's what was studied. Radial threads, web area, web length, all these different parameters were taken into account. These results were all summarised in two principal component axes for web size and silk density. And the map of these phenotypes was separated for each of the different islands. And what they found is when multiple species co-occurred on the same island, the webs of these different species were quite different, probably to avoid competition between different species. However, if you superimpose all these islands on top of each other, then in allopatry you actually get webs which look pretty similar. In the end, it was concluded there are actually only 3 non-unique web types. And if you look at these web types and you relate them to the species of spider that produces them, you can create a phylogeny. And what you see is that the same web forms have evolved multiple times. This provides a pretty big hint that there's strong natural selection acting on these extended phenotypes. So it's very much likely that there's a genetic basis to this. Of course, a lot of our behaviour is determined by our brain, so natural selection acts on brain morphology pretty heavily as well. And this is another thing that's been looked at. Huber et al. In 1997 looked at cichlids. Of course he did. He looked at 109 cichlid species and he found that differences in brain morphology correlated pretty well with the ecology of these cichlids. Now, in Lake Malawi there are 2 types of cichlid. There's the Mbuna cichlids, which are the rock cichlids, and the non-Mbuna cichlids, which are silt, muddy, open water type cichlids. Okay, and Sylvester et al. 2010 did a similar experiment, but this time looking at gene expression. There are certain genes which we know are important in brain morphology, and he wants to see if there was any differential gene expression of these types of genes between these 2 types of cichlid. Because we know that the Mbuna and non-Mbuna cichlids are phylogenetically distinct groups. They form monophyletic groups. Sure enough, it was shown that different genes were being expressed in different parts of the brain, especially these 2 genes here, WNT1 and SIX3. The one thing we've got to take into account is that a cichlid brain can only grow up to a certain size, it can have a certain volume, and that's due to the size of the brain case, which is influenced by loads of other factors. Okay, so that means there's competition between genes in terms of their expression for dominance in the brain. So you get this sort of tug of war going on between genes. And this occurs during development, which as a result leads to different behaviours. Now it's also worth noting that genes don't have to be functional throughout the entire lifetime of an organism. They can be switched on and switch off throughout its life. And these can be influenced through social interactions, which brings us to the very exciting and current topic of Socio Genomics. So this is where social interactions mediate gene expression and behaviour. So brain morphology can lead to differences in individual behaviour, which can then lead to differences in social interactions. And loads of correlations have been made with organisms that do live in a group, and correlations have been made with brain morphology. It's also worth pointing out that this idea of epigenetics can be important as well. This is a relatively new idea where once a protein product is produced, stuff can happen to it after that. Usually it's the addition of things like methyl groups and things like that. And they, as a result, can influence social interactions, influence development, and as a result, behaviour. So let's look at some examples then, shall we? No surprise, we've got some cichlids coming up again, particularly this species, Astatatilapia burtoni. Okay. Now, in this species, the males, the dominant males, are really showy. They've got nice patches of red and blue on them. Okay. The subordinates, on the other hand, aren't as showy. So in a group of these cichlids, there's dominants and subordinates. Now, when an opportunity arises, then obviously a subordinate would like to become a dominant individual, because that means it can breed and have some great fun being the dominant individual. Okay, so once an opportunity arises, a subordinate will go straight in there. And this changes differences in hormone expression. Things like GnRH2, testosterone, and somatostatin. Cortisol is a stress hormone, and also what you see is a decrease in expression of cortisol as well, because as a dominant individual, you're less stressed because the less frustrated you are to not being able to breed. Now, once an individual becomes dominant, it then gets older, it doesn't look as good as it used to, so it then retracts back to becoming a subordinate again. So that's when we see an increase in cortisol levels yet again. Now these guys, Maruska and Fernald in 2010, did some behavioural trials, and they found out that a subordinate shows different behaviour to dominants. Subordinates flee more quickly when you present a stimulus to them, which might scare them, than the dominant. But also, it was found that subordinates can actually become dominant pretty quickly. If a subordinate is isolated, it can become a dominant individual in a matter of minutes. So there definitely seems to be a lot of social suppression of reproduction in these cichlid groups. Okay. Now, the next step then is to look at differences in gene expression, and that's exactly what Burmeister did in 2005. And he noticed differences in expression of this gene, EGR1. Okay. Now, EGR1 is a transcription factor, and it's important in the expression of GnRH2, that hormone that I was talking about. So therefore, it's expressed significantly more in ascending males, and it's involved in the development of the testes. Okay. So that was one study, but then Renatar in 2008 realised, there's bound to be loads of genes. So he took a much more modern approach, did some RNA-seq and microarray analysis to find much more differential gene expression in loads more genes. And these differences in gene expression are affected by differences in sex and social status. And many of the genes identified were important in hormone production, as we talked about before. Okay, so that's a pretty neat experiment. Another experiment done on a different type of fish this time is on swordtails, Cyphophorus nigrensis. And this was a study done by Cummings. And what is nice about this is that it incorporates behaviour and a genomic approach in the same study. So what they did was that they got female swordfish in tanks and presented them with a choice. Okay, these female swordfish were piped up, ready to mate. They wanted to find a mate, right? So they either presented them with a big male and a small male. A big male is presumably a lot better than a small male. Infers better fitness, right? They presented them with two small males. They presented them with two females, which, unless there's any lesbian swordfish out there, I assume they weren't interested in, right? And presented them with no fish at all. And they wanted to see whether there was any differences, any upregulations or downregulations of particular genes whilst these swordfish were making these decisions. And what did they find? Yes, there was. So when these females were faced with a decision, so choosing between a big male and a small male, there were downregulations of specific genes. Okay. When there was no choice to be made, then you don't see any change at all. So you can then make inferences from this about things that we've talked about before. So this seems to imply that mate choice is important in speciation, maybe, due to these differences in gene expression. Yeah. Now, a more detailed analysis was then carried out, looking at 6 particular genes that were known about previously, and what they found was that 5 of the 6 genes that were studied were shown to be important in mate choice, and one of those was EGR1, funnily enough. So you can see if 2 populations have different criteria of mate choice, that means differential gene expression will happen, and that may eventually lead to a speciation event. Okay, enough of fish. The last example is on Polistes wasp, which is an obvious study species for studying social behaviour, of course. And this was studied by Toth et al. In 2010. And they once again did a microarray analysis. So this is when you look at expression of loads of genes at once. And what they wanted to look at was they wanted to see whether there was any differential gene expression between different castes in these Polistes wasp colonies. Because obviously you have queens, you have workers, you have males, and so on. And would you believe it? Yes, there is. 446 out of 3,988 oligos were shown to be very important. And obviously within castes, different individuals do different jobs, so you can also look at differences in gene expression here as well. And what you find is if individuals are doing a similar job in a colony, then they're likely to have more similar gene expression. Okay, so that's a whistle-stop tour of Socio Genomics. We're now going to look at something completely different and see whether traits can evolve which influence habitat choice. Now, there are 4 possible mechanisms in which a habitat preference may evolve at all. So let's go through them. Well, the first one is an obvious one, which is imprinting. Yeah? As you grow up, you learn the environments in which you're living in, and there's no genetic basis to it. You just realise, yeah, I want to stay here. Okay? So you now associate where you grew up with what's a good place to live. Okay? The other mechanism is phenotype dependence. So this is where certain genotypes, which eventually produce a phenotype, are better suited to live in particular areas than others. And as a result of that, these individuals are less likely to disperse. Now, we've got to remember that this isn't about a gene which actually directly affects habitat preference. This just comes about through the loss of individuals which aren't well adapted to this particular environment because they don't have the right genotype. The next one is competition driven. So an individual doesn't actually have to be specifically adapted to a particular environment, as in the second mechanism is suggesting. Instead, certain individuals may just be better competitors. So they're much better at outcompeting other individuals. This can be determined by something else other than being suited to the habitat which you're in, such as body size, for example. Okay, so it's quite different. And the fourth one, which is where we're particularly interested in, is that there's actually a direct genetic habitat preference. So that means there's a gene or a collection of genes out there which directly influences what habitat you prefer to live in. And this has been studied by Whitehead and Crawford in 2006 on another fish, the Atlantic killifish, Fundulus heteroclitus. This species lives in habitats of different temperatures, right? There's a broad temperature range. And what these scientists did is they looked at 327 genes, they kept these fish, and they wanted to measure differences in gene expression with the temperature of the water that those individuals came from. And what they did is that they identified 58 genes which showed expression associated with temperature. However, we've got to be careful here. We've got to correct for phylogeny. That's because individuals which are more related to each other phylogenetically are more likely to show similar traits anyway. So these similarities may have evolved neutrally through genetic drift. Okay? So after you correct for phylogeny, you actually narrow it down to 13 genes. It's also worth noting that when these experiments were carried out, the fish were kept in the same conditions for 2 months, so removing any effects of the environment in which they came from, which may have affected gene expression and so on. Okay. But really, this shows that there's strong natural selection on these 13 genes, which in turn influences how well adapted they are to a particular temperature. So therefore it's easy to understand how natural selection on expressed genes between populations in allopatry could eventually lead to ecological speciation. So there you go then, genes for habitat preference could indeed evolve. That's it for the moment. I'll see you next time.